And do you believe in life after A-levels? Okay, welcome to the Let's Get to the Marks. We're going through the OCR 2023 A-Level Biology Route A Biological Diversity 2015 minutes. Don't forget to start each question, press pause on the video, grab pen, grab paper, have a go, make your revision active, and then unpause and watch the explanation. All right, question one. Which option correctly describes a tissue? A group of cells performing similar functions, a group of organs performing a specific function, a group of similar cells performing a specific function, a group of similar cells performing a variety of functions. If you don't know the answer to this, you need to bust out your revision cards, write down the question, what is a tissue, and then get the answer there. So it's a group of similar cells. So we're looking at C and D, because it's definitely a group of similar cells, and it's not a group of organs. Tissues make up organs. So a group of similar cells performing a specific function. So for instance, muscle tissue contracts. That would be the example that I would keep in my head so that you can test yourself and work out what a tissue is. Muscle tissue contracts, elastic tissue recoils, etc. Right. Bone marrow stem cells are a source of erythrocytes. That's red blood cells. Which statement about bone marrow stem cells and their role in the production of erythrocytes is correct? A. Bone marrow stem cells are totipotent and can therefore differentiate into any type of cell. Erythrocytes are the only blood cell produced from bone marrow stem cells. Erythrocytes need to be replaced from bone marrow stem cells because erythrocytes are unable to undergo mitosis. D. In humans, differentiation of bone marrow stem cells into erythrocytes involves the synthesis of a haploid nucleus. So which one of these statements is a true statement? Pause the video, have a go. Okay, I'm going to go through the explanation. Right, totipotent, pluripotent, multipotent. Make sure you know the meaning of totipotent, multipotent, and pluripotent when it comes to stem cells. If you don't know that, get it down on a revision card. Bone marrow stem cells are totipotent. They can differentiate into any type of cell. No, that's not true. If bone marrow stem cells could do that, then if you had your arm chopped off, you could grow a new arm, couldn't you? And people wouldn't be paralyzed if they could grow new spinal tissue. So we can tell, obviously, that this one is not true. I mean, if that was true, you could grow a new head or something. Right. Erythrocytes are the only blood cell produced from bone marrow stem cells. What about that one? Red blood cells are the only ones made from bone marrow stem cells. You might remember when you study the immune system that you have something called white blood cells. And you might remember two of the types of white blood cell. And you have T cells and you have B cells. And the reason why some white blood cells are called B cells is because they're produced by the bone marrow and some are produced by the thymus gland. So they are called T cells. So no, you can make white blood cells as well from bone marrow stem cells. So this is incorrect. Arsehole. Erythrocytes need to be replaced from bone marrow stem cells because erythrocytes are unable to undergo mitosis. Now, you might not have been taught this specifically, but what is it that erythrocytes or red blood cells are missing? And hopefully you know erythrocytes have no nucleus. And because they have no nucleus, they don't have many organelles, especially we need to know they don't have a nucleus. Therefore, how could they do mitosis? They don't have spindle fibers. They don't have a nucleus. So you can't do nucleus division. You can't do that if you don't have a nucleus. So yeah, that is definitely correct. This is the true statement. This is correct. The answer here, folks, is C. I won't bother going through that, but yeah, you only get haploid nucleus inside sperm or egg cells, so nothing to do with red blood cells there. All right, let's go. Question 3. Which option correctly describes the sequence of events during the cell cycle? Why don't you see if you can draw the cell cycle out before you answer this? If you can draw and you know it off by heart, this is just a straight content memory recall. Do you know the cell cycle off by heart? Draw it out, pause the video, have a go, and then try and work out the answer. Unpause, I'll go through the explanation. So I'm going to draw it out for you. Right, we've got our cell cycle. So our cell cycle, oh my days. What do we have for the cell cycle? So we've got it doesn't really matter where I start. So I've got mitosis, cytokinesis, then I'm going to go gap phase 1, then I'm going to go S for the synthesis phase, and then gap phase 2. There's kind of 5 parts to the cell cycle, and we're going around like that. It doesn't really matter where you start. You can start with cytokinesis, start with G1, start with this, but essentially we need to be in this order, okay? So let's go. Cytokinesis, that's good. Then the interphase. Oh, by the way, we can call all of G1, G2, and S can all be known as the interphase. So you have to be able to think on your feet because so does this make sense? Cytokinesis followed by the interphase, then G1, G2, S. No, they're in the wrong order there. And the interphase is G1, G2, and S. So definitely A is not your correct answer. Then let's look at this. Cytokinesis followed by mitosis. No, cytokinesis, then interphase, then mitosis. If you want to do that, it should be cytokinesis, interphase, mitosis. That is wrong. And then what about this one? G2. Let's start with G2 here, just in case you're wondering. That there is supposed to be a 2. So let's start with G2. G2, mitosis, cytokinesis. Yep, this is going good. G1, then S. That is correct. That's the correct order. So once you can draw this cycle out, you can't really get this question wrong because you can just check the order off. And then this one, the reason why D is wrong, we go mitosis. It's saying then the interphase, then cytokinesis. No, that's wrong. Cytokinesis comes after mitosis and before the interphase. So this one is wrongy wrong. All right, nice one. Let's move on. Keep moving on, keep moving on. Right, scientists crossed 2 plants that were heterozygous for 2 different genes. Both plants had the same genotype, capital A, little a, big B, little b. The genotypes and ratios of the offspring resulting from this cross were AABB1, AABB2, and AABB1. Which option could explain these observations? So a little bit of knowledge. This is dihybrid inheritance where you're getting 2 different genes. It's not monohybrid inheritance like where you do brown eyes, blue eyes, one phenotype. We've got 2 different phenotypes here. So big A and little a might be for one type of gene, I don't know, hair color, and big B and little b might be eye color, whatever. 2 genes that are connected, and we call that dihybrid inheritance. You get 16 possible offspring, and they've got this in the ratio of 1 to 1, so it's like 4, 8, 4. Which option could explain these observations? Pause the video, have a go. Right. Crossing over just explains how genes get swapped when you form the chiasmata and alleles are swapped. So this is not because of crossing over. That's how you get variation. Epistasis, you might think, oh, I think this could be something to do with epistasis. You might remember something about epistasis. Epistasis is really where one type of gene, you get dominant epistasis, which would be like the big capital A, basically masks the expression of the B. So that gene is not expressed when this is present, or the capital B, when that is present, the dominant allele for B is present, A doesn't get expressed. So that would be dominant epistasis. You get recessive epistasis. You get recessive epistasis, which is where if two of the recessive alleles are present, they would mask the they would mask the expression of the other gene. Now, that's really about phenotypes rather than the genotype ratio. This would affect the phenotypic ratio that we would see. So I'm not going to go with epistasis. Mutations, they're not going to explain that. That's just a change in the nucleotide base sequence. That's not going to explain that. So we're left with autosomal linkage. And you tend to get autosomal linkage when you don't get the right amount of different crosses. Like, look, we're missing here. What are we missing? If you were crossing a big B, little b, oh my gosh, what am I on about? And we were doing this two heterozygotes, and we're crossing them. Where is the little a? We haven't got a little a, big B, big. We haven't got that there. There's quite a few that are missing combinations, and this is going to be because of autosomal linkage. It's where some of the alleles are very close to each other and cannot be separated, so that you can't get some of these combinations. Okay, let's move on. The student was observing and measuring cells using a light microscope. Which option describes a method that would enable the student to accurately measure length of a cell? Why don't you pause the video, have a go at this one? Right, I'm going to go through the answers because I'm taking too long. View the cells using a calibrated eyepiece graticule and note the magnification of the eyepiece lens. View the cell using a calibrated eyepiece graticule, note the magnification of the objective lens. View the cells using a stage micrometer and note the magnification of the eyepiece lens. View the cells using a stage micrometer and note the magnification of the objective lens. Right, the stage micrometer or micrometer is used to calibrate the graticule. Once you've calibrated, so the stage micrometer, you've got your microscope that you're having a little look down, and you put your stage micrometer on there. This is your stage micrometer, okay? And then you attach an eyepiece graticule that basically has its own lines on it that are going to appear down here, and you use this to work out how much each division is on your graticule. Once you've calibrated your graticule, you remove the stage micrometer, the ruler thing, which is on a slide, and then you replace it with your slide with the actual specimen, whatever it is that you want to look at. This has now been calibrated, and you know how much each division is in your graticule. So what do we have to know here? View the cells using a calibrated eyepiece graticule, and you'll need to know the magnification of the objective lens, like whether it's times 40, times 50. So the answer here is going to be B. Wunderbar. Excusez-moi. Question 6. Which of the following describes from lowest to highest the resolution of images that can be achieved by different types of microscope? So what do we have here? We have light microscope, scanning electron microscope, transmission electron microscope. So we want to go from low to high. Now, light microscopes have the lowest resolution of all because they are limited by the width of a beam of light, which is roughly 200 nanometers. I don't know how wide an electron is, but electron microscopes have a much higher resolution because electron microscopes are just the width of a single electron, which I guess is way smaller than 200 nanometers. So anything which doesn't start with a light microscope is wrong. So it's going to be A, B, or C. Now we go light microscope, then the scanning, then the transmission. I like that, and that's the correct order. The highest resolution is the transmission electron microscope, which passes through the specimen. The scanning electron microscope uses bombardment, which bounces off the specimen, and then the scattering pattern of the electrons is used to create a false color 3D image. So I am going with A here, folks. That is the correct order. It goes light microscope, then you'll have you notice they mentioned laser scanning confocal. So it's going to go light microscope, then the laser scanning confocal, which has a higher resolution than the light microscope. And then after that, the scanning electron microscope. Remember, the laser is still using light. The L in light, I'm pretty sure stands for, in laser, sorry, stands for light. So that's still going to be limited by the width of a beam of light. Okay, let's zing on. Oh my gosh, got a dry throat. Lactose metabolism in E. coli is controlled by the lac operon. Which of the structures associated with the lac operon contain nucleic acids? 1, the operator. 2, the regulator gene. 3, the structural gene product. So remember, you probably remember a stretch of DNA known as the lac operon. So we have like a, what do we have? We have like, oh my gosh, operon. We have the operator, we have the promoter, then we have like X, Y, Z, whatever. Anywho, I don't know what I'm going on about. Probably got ROP. What the hell is that? Okay. All of this is part of DNA. This is all DNA. So the operator and the promoter and the regulator gene, this one here, which actually creates the protein that binds here. So all of this is going to be made of nucleic acid. So definitely 2 and definitely 1, the operator. They're all found on the, what do you call it, on the DNA. The structural gene product, what is the product of a gene? It's a protein, and proteins are made of amino acids, not nuclear acids. So we're getting rid of 3. 3 is wrong, so we're going to go 1 and 2 only. The answer is B. Okay, I don't think I explained that very well, but there we go. Essentially, 3 is a protein because it's a product of a gene. So it's not going to be made of nucleic acids. The others are all found on the DNA. Homeobox genes control the body plan in eukaryotes. They're the things that make sure you have 2 arms, 2 legs, etc., and 1 head, blah, blah. Which of the statements about homeobox genes are correct? They bind to transcription factors. They contain a highly conserved sequence of 180 base pairs. They regulate the expression of structural genes. So why don't you pause the video and have a go? Okay, homeobox genes do not bind to transcription factors. They contain a highly conserved sequence of 180 base pairs. That is true. We find that they are conserved throughout all organisms, or most organisms. Most animals have the same stretch of homeobox genes. Okay, they regulate the expression of structural genes. This is true. So I'm going to go 2 and 3. We're going to go C with this one. Sure, I could have done a better explanation of that, but try speedy. Okay, the graph below shows the population of viable bacteria in a flask over 48 hours, which time A to D has the highest bacterial death rate. So when is the most bacteria dying? Look, it's for sure there are more bacteria dying than being born. Here, there are more bacteria being born or being produced, however you want to say it. They don't, yeah, anyway, there's more bacteria being born. The birth rate is higher than the death rate here. There are bacteria dying here, even though their numbers are going up, but the birth rate is far exceeding the death rate. Here, the birth rate and death rate are equal, and the number of bacteria is maintaining. But this must have the highest birth rate and the highest death rate because the population is so high. So we're going to go B. So that has the highest bacterial death rate. Although here, of course, the birth rate is dropping due to lack of resources, etc. And we know it can't be C or D because their gradient is the same, so their death rate is pretty much maybe potentially equal. Okay. Shrinky. Oh, shit. Excuse me. Right. The image below shows representations of structures of 4 polysaccharides. We've got amylopectin, cellulose, amylose, and glycogen. Use the image to answer questions 10 and 11. Which of the molecules contains the highest proportion of 1 to 6 glycosidic bonds? Is it amylopectin, amylose, cellulose, or glycogen? Give your answer. Pause the video, have a go. Okay, it should be really easy to visually see. This one has the most branching. Branches are found on the 1 to 6 glycosidic bonds. 1 to 4 is the straight chain here, or all of these are 1 to 4. All of these are 1 to 4. There's no branching in this, so these only have 1 to 4. This has 1 to 6. Wherever there's a branch, it's a 1 to 6 glycosidic link. And you can see here there's far more branching, so the answer is D. That has the most glycosidic 1 to 6 linkages. Let's go. Right, question 11. Which of the molecules is not used for energy storage? We don't really need to bust back up and have a look at this because you should know this off by heart anyway. And the answer is cellulose. Well, it's not used for energy storage. It's a structural molecule used in the cell wall of plants. Okay, let's move on. Right, question 12. The modern pedigree dog breeds have been produced by many years of selective breeding. Which option does not explain why genetic disorders are common in pedigree dogs? Characteristics that are desirable to humans can be associated with features that are harmful to dogs. Selective breeding increases the chance of an individual being homozygous for certain characteristics. C. Selective breeding increases the chance of mutations. Selective breeding is associated with inbreeding depression. Which option does not explain why genetic disorders are common? Okay, so yeah, mutation. When you think of the word mutation, you should think of the word random. So random means it's not affected by temperature. It's not affected. The rate of mutations is a random thing. It's not affected by these external factors like doing selective breeding. That doesn't affect random mutations. However, selective breeding, of course, reduces the gene pool and increases the chance of inherited genetic disorders. And that is, we can go with all of these as possibly being true for the others. So we're going to say C here. Okay, let's have a look at number 13. Which step could be taken to reduce the occurrence of genetic disorders in pedigree dogs? Breeding only with individuals that meet official breed descriptions. So that's kind of like inbreeding because that's a limited amount of variety. Broadening the official descriptions of pedigree dogs, that will definitely reduce the occurrence of genetic disorders because you're basically, if you're saying your dogs can only be of a certain height, like Chihuahuas can only be, I don't know, below a certain mass or whatever, you're going to exclude some of the gene pool when it comes to breeding, and you're going to reduce the amount, the gene pool. So this is going to widen the gene pool by allowing other variety or dogs with different alleles in. Increased use of somatic gene therapy, no. Regular crossbreeding with wild ancestral domestic dogs, no. Right, it's going to be B, by widening the gene pool. 14, organisms are named using the binomial system. What is the correct way to represent the binomial name for domestic dogs? So this is really testing you on, do you know this? Why don't you pause the video, have a go at this. Now, the first name is the genus and begins with a Capital. So we're looking for the one that begins with a capital. So it can't be C or D. The first name is always the genus and it always begins with a capital. The second word or the second name um, is the species and is in italics or lowercase, no capitals. So it depends whether it's typed or what, but it's in italics or lowercase. So we're going to go with this one. This one's got the funky kind of writing. And so we're going to go with A, Canis familiaris. And the difference there is that's just lowercase. It's not in italics. Okay. So A is in italics. That's why we're going for A. Okay. I think there's one more question. It's the last one. Okay, the image below shows an enzyme and its substrate. Which option represents a coenzyme for this enzyme? So which of these will enable this to fit better into that active site? Why don't you pause the video, have a go? Bit of a spatial awareness one. Oh, look, straight away, if you take this triangle and you add this triangle here, guess what? It is going to make that fit so much better. Look, that now fits perfectly. Unless you either add it on there or it locks in there and then that fits perfectly. It makes a better fit for the substrate into the active site, a more snug fit, in which case this breaks down faster. So that is going to be the coenzyme is going to be A. All right, guys. That's the end of the MCQ for the 2023 Paper 2 Biology. Yeah, don't forget sharing, caring, hit the like, punch up the bell, subscribe. Let's get to the marks. I'll catch you lot on the next video. Okay, 16. Many organisms undergo asexual reproduction by mitosis. State two other roles of mitosis in organisms. B. Plants reproduce asexually by mitosis. Explain why mitosis and not meiosis is used for asexual reproduction in plants. And then we have 2. Bacteria reproduce asexually by a process called binary fission. Suggest why bacteria are unable to reproduce asexually by mitosis. Why don't you pause the video, have a go at these roles. So reproduction is one of the roles of mitosis, but of course we have growth. So when you're growing new hair, new nails, and you want identical cells, new skin, you do that. And then repair, like when you cut yourself and you need to heal that, or you need to grow new tissue, you've got repair old tissue, or grow new tissue. So growth and repair. We've then got plants reproduce asexually by mitosis. Explain why mitosis and not meiosis used for asexual reproduction in plants. This is a bit of a strange one, but essentially because some plants do use sexual reproduction and some use asexual reproduction. Of course, the ones that use sexual reproduction produce gametes and do use meiosis, so the birds and the bees, etc. However, let's get on to why we bother using mitosis. Well, what's the advantage with mitosis is you get identical offspring produced. So identical offspring produced. Now, that's not necessarily always an advantage, but it is if you've got some good genes that you want to be guaranteed in the offspring. So that is one of the advantages of mitosis. You get, and I want to just add this in because it's A level. I want to say genetically identical offspring produce because, of course, they're going to have different environmental factors, and that means that they won't actually be identical phenotypically. They're going to like have more water, more nutrients, more sunlight, etc., depending on where they're growing up. So genetically identical offspring produced, and also mitosis ensures the diploid number is maintained. So ensures that the diploid number is maintained. And there we go. Well, what do we mean by that? Well, let's say your diploid number is 14. Then if you copy yourself identically by mitosis, you're going to make the new cells will also be 14. The zygotes will be 14 or the new cells will be 14. Whereas, of course, not zygotes. Remove that. If you do meiosis, you're going to half that number when forming the gametes. So you're going to have 7 in your male and 7 in your female gamete. Now, of course, fertilization brings them back and restores the diploid number, but that's the longness of sexual reproduction. You've got to restore the diploid number through fertilization. Okay, so we don't have to do that with mitosis. One of the advantages is that the diploid number is maintained. No need for fertilization. Okay, bacteria reproduce asexually by a process called binary fission, slightly different than mitosis. Suggest why bacteria are unable to reproduce asexually by mitosis. So let's think about, where does mitosis happen? Mitosis happens in the nucleus. What do you know about prokaryotes like bacteria and the nucleus? Well, quite simply, they do not have a nucleus. So we want to say mitosis occurs in the nucleus and bacteria do not have a nucleus. So therefore, they ain't doing no mitosis. Bosh. Right, let's move on. Oh my gosh, if this thing will let me actually move on. Right. Oh, yeah, 6 marker. That's not what I wanted to see. C, asexual reproduction, also known as cloning. Many organisms can be artificially cloned for commercial purposes. Discuss the arguments for and against artificial cloning in plants and animals. Notice these. This is really key for this question. So grab your paper and your pen. Notice these and. So if you want to attempt to get 5 or 6 marks, then you need to do the arguments for and against. So plus and minus, and you need to do plants and animals. So the way I break it down is, what's a positive about cloning in plants and a negative about cloning in plants? What's a positive about cloning in animals, and what's a negative about cloning in animals? Now, there might be some crossover where the negatives are the same. Anywho, let's go on and smash that out. Okay, so let's go through some generic advantages. So for the advantages, which I can't spell, so be an advantage if I could spell. Advantages are that you can produce large numbers of individuals rapidly. Um, yeah, that's for both animals and plants via cloning. Also, the offspring, this is for both, by the way, animals and plants, will be genetically identical. So you can guarantee desired traits. What do I mean by that? If you've got some really juicy strawberries with the juicy strawberry gene, we can guarantee that the offspring will also have the juicy strawberry gene. So that would be the advantage with plants. And of course, the advantage if you have chickens that high lay egg layers, they lay lots of eggs, and there's a gene for that, and you clone them, then the offspring are also going to have the gene for being high volume egg layers or something like that. So you're guaranteed that desired trait. I don't know why I've called it the desired traity, but yeah, you get that. Right. So that's going to get us a couple of marks for some nice advantages there. What are some other advantages? Right. Endangered animals like, I don't know, the panda. If you clone the panda, then you're more likely to keep them alive by having copies of it. Endangered plants, certain orchids, you could clone them and get copies of them. Also, cloning skips out the whole fertilization and mating and all of that sexual reproduction, especially with animals. You don't need to find a mate. So you could actually conserve the species through cloning. So let's put conservation of a rare species of animal or plant through cloning. I mean, some people clone their pets. They pay money and have a clone of their pet made, so they get a new dog and it's identical to their old dog, and they seem to like that. That seems a bit weird, but there you go. So conservation of a rare species of animal or plant through cloning, that's another positive. Should we go into the negatives? We could do some of the against. I think I've done three kind of positives: large amounts of individuals, genetically identical, so you guarantee the desired trait, conservation of a rare species of animal or plant. Animals can be cloned to produce identical pets for owners. I know it seems a bit weird, but we'll give that one as an example. Like if you've lost your cat and you want little Harry back, you can clone them. And for plants, you've all eaten bananas. They're seedless plants. Wild bananas have big stones in them, basically massive hard seeds, and are really hard to eat and pretty much inedible in that way, certainly on a mass scale. So we've cloned bananas. Any banana you eat in the shops is actually a clone, and it is a clone of a seedless version of the banana plant. So for plants, cloning seedless varieties. Varieties. Oh my gosh, cheese varieties. I can't. So I'm just going to put that for plants cloning seedless varieties. That would be an advantage. E.g., I can't spell bananas. Is that how you spell bananas? E.g., bananas. What else? What else? What else? What else? Oh, we haven't done all the bad disadvantages. I've definitely covered enough positives. Let's hit you with some disadvantages. So disadvantages, obviously, reduction in the gene pool, loss of alleles. So what essentially we mean there is by only cloning, you're not really getting the introduction of other genes. You're reducing the size of the gene pool. And I guess on the same side, you could say less biodiversity. When you do cloning, you're going to get less biodiversity. Now, what's the problem with that? This means the population is less able to adapt to changes or sudden changes in the environment. So it's more susceptible to becoming extinct, essentially, less able to adapt to new diseases or changes in the global temperature and things like that because of the loss of those alleles and the small gene pool. What else can we go? What else can we go? What else can we go? Yes, implants, cloning may, uh, involve offspring um, contaminated with viruses from parent. So there might be some sort of virus that is contaminated from the parent. What else? What else? What else? Somatic cell nuclear transfer, SCNT, is not very successful, is not efficient, and offspring often have health issues. If you remember Dolly the sheep, the first one, I mean, they die very young, don't live very long, healthy lives. So those would be some of my disadvantages, those ones at the bottom here. So these are I seem to have a lot of advantages of cloning over disadvantages, but I don't want to go on forever. So, oh, that's a disadvantage there. Let's get rid of that. And so these are all the disadvantages down here, and these are all the advantages, and that will get you 6 marks. Six, 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 six. Okay, enough of that. Let's go. Oh my gosh, this question doesn't stop. D. During meiosis, the independent assortment of chromosomes leads to the formation of genetically different gametes. Gametes of the western clawed frog contain 10 chromosomes. Gonna need my calculator here, I can sense it. Where's my calc? Oh my gosh. Oh, it's right in front of me. Right. Calculate the number of genetically different gametes resulting from the independent assortment of chromosomes that would be produced by an identical frog. So this is long. This is long. This is long. The gametes have 10 chromosomes, which means when you assault these bad boys, you've got 10 in the sperm. So let's call this gamete 1, 2, 3. Let's call this chromosome 1, 2, 3, 4, 5, 6, 7. This is long. What the hell am I doing? 9. Oh my gosh. I'm just showing you what the issue is and that there's a simple mathematical way to do this, but really I'm not 100% sure why I'm doing this. This is just to show you what we're dealing with because you might actually not know what they're asking you. You might think, what the hell are they asking you? Number of different gametes. Okay, so how many different gametes can be made Oh my gosh, what happened there? Yeah, that's supposed to be like that, and then 10. Okay, so you've got 10 different chromosomes from mum, 10 from dad. Calculate the number of genetically different gametes resulting from the independent assortment of chromosomes that can be produced by an individual frog. So this is my individual frog's chromosome, and let's just say we're picking on a man, okay? He wants to make a sperm cell. Let's call him Bill the frog. Bill the frog wants to make a sperm cell. How many different combinations? He has to have a 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, but he could have like the black one, blue two, or he could have a black one, black two, blue three. He's got to have all 10, only one of each, but it can be any combination of the black or blue. So how are you going to work out mathematically how many different combinations of all 10 he can have, bearing in mind that he can have all 10 black, all 10 blue, or any combination of black or blue, i.e. male or female from his mum or his dad when he makes his sperm. So how many different gametes can he make? Why don't you pause the video and have a go? Okay, so we're looking at 2 to the power of 10. What does that equal? Let's smash that in the calcs. So let's go. Oh my gosh. Oh my gosh. It's 1,000. Oh, I've done it. 1,024. That took me a long time. I think I'll have to speed this one up. I'm going to put this one on speed talk. So I'm going to be talking like when you watch this through on a video because I'm recording this slow and it's taking far too long. Okay, let's go. Okay, question 17. Catalase is found in many tissues. It is an enzyme that breaks down hydrogen peroxide into oxygen and water. Some students do an experiment to investigate the effect of hydrogen peroxide concentration on the rate of reaction using the apparatus shown in figure 17.1. You can see here we've got a bung, we've got a conical flask, there's a potato cylinder in there, which the potato cylinder itself is a source of catalase and hydrogen peroxide. Well, actually, it's a potato cylinder sitting in a solution of hydrogen peroxide, H2O2, and the potato is the source of catalase. Right, we've got a measuring with delivery tube, so any gases that are released can come down here. We have a measuring cylinder here to measure the volume of the gas, and we have a water trough. Okie dokie, this is the method used? No, this is the method the students use. Use a stock solution of 250 centimeter cubed of 20 arbitrary units, can't say that word, hydrogen peroxide to make solutions of 8 AU and 2 AU. Cut 15 cylinders of potato equal diameter using a cork borer and use a ruler to ensure that each one is 5 centimeters long. 3. Set up the apparatus as shown in 17.1. Record the starting position of the water in the measuring cylinder. Place one cylinder of potato into the conical flask. Pour 50 centimeter cubed of 2 AU hydrogen peroxide into the flask. Immediately secure the bung and start a stopwatch. Record the volume of gas given off every 30 seconds for 3 minutes. Repeat steps 3 to 7 twice. Repeat steps 3 to 8 using 8 AU and 20 AU hydrogen peroxide. State the independent variable in this investigation. And then after you've stated the independent variable for 2 marks, describe how a student could produce an 8 AU solution from a 20 AU stock solution. Why don't you press pause on the video and have a go? Well, I'm going to go through the answers. So when you want to find what the independent variable is independent of the investigation. That means it doesn't change via the action of the experiment itself. It's independent of the experiment. It depends on the investigator. So you change it. So the independent variable is what the scientist changes. The dependent variable would be the volume of oxygen given off because that is dependent on the reaction, the actual chemistry or biology that's going on. What's independent of the investigation is down here right at the end is the concentration of hydrogen peroxide. That is not being controlled by the investigation. That's being controlled by the scientists. So we want to say hydrogen peroxide concentration for that first one, and that will get you the mark. So limit that in hydrogen peroxide concentration. And that's in AU, which just means they haven't given it any proper units. So describe how a student could produce an 8 AU solution from a 20 AU solution. So what you want to do here is just explain the maths of how you would water something down, basically, so that it is 8 out of 20. So what is 8 out of 20? Well, 8 over 20, where's my calc? What is 8 over 20? What's that, 60%? So no, 40%. So that's going to be 0.4. So you basically want something to be 40% as strong. So what you're going to do here is, depending on the volume you want a 2 to 3 ratio. So you want like a 2 to 3 ratio of water, of hydrogen peroxide, 20 AU to water. And of course, you want to use distilled water. And if you want to put that into amounts, let's say you wanted to produce 10 centimeters, then I would go 4 centimeters of cubed of hydrogen peroxide, which has the formula H2O2. You don't need to know that, by the way, to 6 centimeters of H2O. And that is going to get you a 40% strength of the original solution, right? Because you're only using 4 centimeters of the original solution and 6 centimeters of water. So it's 40% of 20, which is 8. Cool. Or if you preferred, you could do it out of 150. So you'd use 100, you could do 250. So 100 centimeters of the H2O2 hydrogen peroxide. And you could do that with 150 centimeters of water. So there's just another example of how you break it down. You could do 40 centimeters of hydrogen peroxide and 60 centimeters of water if you want to make 100 centimeters of stock solution. They didn't give us a volume, so you can give the examiner examples if you want, or you can just show the calculation and show how you came out. It's a 2 to 3 ratio. Beautiful. Okay, let's quickly mark for that as well. Let's move on. Liver tissue contains a higher concentration of catalase than potato. So it's more concentrated catalase enzyme in liver than in potato. So just two reasons why the student chose potato rather than liver. So the whole point here, the only reason to choose potato rather than liver is because it has a smaller concentration of the enzyme. So this has less enzyme, right? So why would you use the potato with less enzyme? What could possibly be the reason? It must be that if we use the liver, which has more enzyme, it's going to happen too fast. And why is that a problem? Well, it's harder to record all the details of a reaction if it happens too fast. So we want to say with liver, reaction happens too quickly. Now, what's the problem with that? It might be difficult to record. Maybe it would, with liver, the reaction happens too quickly is over too soon. I don't know if that will get us a mark. That seems a bit thing. Violent frothing, violent bubbling, or frothing could like dislodge equipment, or make it difficult to read measuring cylinder. So it could like bubble up in the measuring cylinder, dislodge the delivery tube. It could feel froth from the liver up in there. If it happens too quickly and it's over too soon, I think they were trying to do every 30 seconds for 3 minutes. Well, if it's over in a minute, then you can't do that, can you? So all of these could be viable answers. But yeah, essentially you should be able to work out, but it's because it's going to be too quick because this one has less enzyme and that one has more enzyme. So that rate of reaction is going to happen too fast. You're going to get more error in that. The potato cylinders were cut to equal length, suggests a further precaution the students should have taken when preparing them to ensure the investigation was valid. So you've got your potato, and essentially they've been cutting cylinders out of the potato. What should they have kept the same about the cylinders of potato? They cut them to equal lengths. What we want to do, I mean, the width was controlled by the potato borer, the thing that cuts into it. I would say the species of the potato, the species or variety of potato. They might say variety rather than species. So it's like a subspecies. So the species or variety of potato. What else do you want to keep the same? Make sure you have peeled the potato. You really got to check the mark scheme for this one because some of the things you might assume, like I guess the age, how old the age of the potato, older potatoes may have less of the enzyme, something like that in them. Okay, let's give ourselves a mark on that. Okay, let's move on. Oh gosh, it looks like some calculations. Some of the students' results are shown in the tables below. So we've got the results for the 2Au hydrogen peroxide. That means there's a low concentration of hydrogen peroxide. We've got the time taken. We got repeats, 1, 2, 3. We've calculated a mean, and we've got the standard deviation around the mean. So that's just the lowest to the highest. Let's zing down. 8Au, we've got 8Au, so a slightly higher concentration of hydrogen peroxide. And then we've got the results for 20Au, which is a much higher concentration of hydrogen peroxide. Of course, that's our substrate, and the catalase is the enzyme. Calculate the standard deviation. Ooh, got the equation there. Let's zoom in and make this a bit big for you, man. No, did he? Calculate the standard deviation for the 60-second result at 20Au hydrogen peroxide. Use the formula S equals what we've got the square root of the sum of x minus the mean squared over the total number of individual values minus 1, and that will give you the standard deviation. And we know it's at 60 seconds and at 20 AU. Let's check that one out there. It's 2 marks. So you're going to get 1 mark for calculating the mean, 1 mark for calculating your standard deviation. So we're looking at 60 seconds, and we're on this one here, which I've just got on the screen. So we want 60 seconds, and we need to calculate up. Oh, hold on. Hold on, Governor. What's the mean? The mean is 76.7, and we need to calculate the standard deviation. Okay, and then we can put that in there. So that's where we're going to put our answer, although to be honest, you can just write it down here. So 60 seconds, and our mean is 76.7. So we need to set this out like this, square root, and we need to do the sum of x minus the mean. So we're going to take the first result, which is, why am I using pink? That's the first question to ask. 78 minus 76.7. I want to square that, and then because we're doing the sum, I want to add these all together, obviously. I want to do 74 minus 76.7. You might say, why do you square it and then square root it? Aren't you cancelling it out? Well, by squaring it, you get rid of negative numbers. Like that's going to be a negative number. Doing a negative times a negative, you get a positive, and then you square root to get rid of the root after you've squared them all individually. So that's essentially why they do that. And then we're going to do 78. That's going to be the same answer, isn't it? Minus 76.7. We're going to square root there. And then that is going to be our x minus the mean, which is this, x minus the mean squared. So we're going to get that by adding that all together. And then we're going to divide that by n minus 1. And n minus 1 is 3 because there are 3 different values. And then we're going to minus 1. So 3 different values, minus 1. We basically divide it by 2, and that should get us our answer. Alrighty then, let's have a go. Smash it in the calc. Okay, so we got 3 minus 1 for n. What do we want? We want to add all of this stuff up. So if I square root that, I'm just going to simplify all of that. Let me switch to black. 1.69. And then that's what I get for that. Squaring that, then it's 74 minus 76.7. I'm going to get 7.29. That's what you should get in your brackets for that. And then 1.69 for that. Now, you're not going to get any marks for doing all of that. Sum all of that stuff, guys. And if you sum all of that, you will pick up 1 mark for working out that you're on top of the line. What's that called? Denominator, numerator, numerator. Oh, God knows. 10.67. So for working out what's up there, you're going to get 10.67. That will score you a mark. Remember, we still got to square root this bad boy. And then 3 minus 1 for our n, which is our number of values. We're going to have 2. And if we divide that by that, let's smash that in the calc. What is 10.67 divided by 2 equals 5.335. But we still need to square root it. 5.335, square root that bad boy. And if we square root that, we're going to get something like 2. Square root answer, please, Bob. 2.309, I guess 2.31. So I'm going to get like 2.3 or 2.31 for that one. So there we go. All righty then. Sorry about that. Just had to take a phone call. Anyway, so what do you get the marks for this standard deviation? Well, 10.67 will get you a mark. Maybe for working out that you're supposed to do this with the mean, showing some of the working out and then seeing 76.7 being taken away. That might get you a mark, but definitely 10.67 will get you a mark. And then I guess 5.335 should get you a mark because it's just showing some of the working. And of course, the final answer will get you both marks. 2.3. It was 2.309, so you could round it to 2.313 sig fig. Doesn't really matter. And that's going to get you both of those marks. Let's move on. And that's for the standard deviation. So 2.3, and you do your kind of common sense check and go, yeah, that looks good. Right, let's zing. Use the standard deviations to discuss the repeatability of the student's results. So if we check the standard deviations and we check the repeatability, let's look at 20 AU, the standard deviation is quite high. But when we look at this table of results, 8 AU, look at the standard deviations. So at the lower concentration of 8 AU, the standard deviations are lower. And if we go to an even lower standard deviation, concentration, what am I on about? 2 AU, so a lower concentration of substrate of hydrogen peroxide, we've got an even lower standard deviation. So the first comment is to say that as you decrease the hydrogen peroxide concentration or substrate concentration, the standard deviation is lower. It's more repeatable. But also that as time goes on, we can see with all of the concentrations, as time goes on, the standard deviation increases, so it becomes less repeatable. So that's what I want to put down. Can I remember it by the time I get to the rating? Probably not. So let's go at lower concentrations of hydrogen peroxide. Oh my gosh, check out that spelling, hydrogen peroxide. The SDs decrease, and so the it is more repeatable at lower concentrations. And then the second thing we noticed with all concentrations, at all concentrations, the results are more repeatable at small. Oh my gosh, what's happened to my writing? So at all concentrations. Jeez, this is painful to watch. I'm going to put this on high speed, so don't be surprised if you're listening to this because it's just taking too long for me to type out. At all concentrations, the results are more repeatable at lower times. Repeatable at lower times. Yeah, as they have smaller standard deviations. Okay, I think we got all the information there. Basically, the lower the concentration, the more repeatable, and also early on in the experiment, it is more repeatable. Okay. The students calculated the initial rate of reaction over the first 30 seconds. Calculate the rate of reaction over 30 seconds for the result at 2 AU hydrogen peroxide. I'm going to go up to the table so we can see it. So we want the rate of reaction 30 seconds 2 AU. Let's boom. So let's go here. Why don't you pause the video and have a go? So you can see 30 seconds and the mean is 5.3 centimeter cubed produced. So I just want to do 5.3 divided by 30 seconds. That's centimeter cubed. So let's smash that in the calculator. Should be nice and easy, folks. Should be a dead easy mark here. 5.3 divided by 30. And I have 0.176 equals 0.176, and that is going to be centimeter cubed per second. So centimeter cubed per second, you're going to get a mark for the units. So I'm doing it in centimeter cubed per second. Now, if you want to change that to per minute, you could go 5.3 divided by 0.5, and then you can find out how many centimeters cubed per minute. So it's going to be roughly half that per minute. No, double that per minute. What is that going to be? 10.6 per minute. Okay, let's zing on. Okay. Here we go. When analyzing the results, the students assumed the volume of oxygen collected was the same as the volume of oxygen produced from the breakdown of hydrogen peroxide. Suggest 2 reasons why the value the students recorded for the volume of oxygen might not accurately reflect the volume of oxygen produced. So they made the assumption that as the gas entered the measuring cylinder, this was equal to the amount of oxygen released by the hydrogen peroxide. Why might this have been a mistake? Let's check out the apparatus. Where's the apparatus? Oh my gosh, right up here. Okay, so if we check, let's zoom in on this. So if we check out this apparatus here, guys, then what the question's saying is they assume that the volume in there is all the oxygen given out from here, is equal to the oxygen giving out from here, that this is oxygen. Well, for a start, there might be other gases in the water or in the delivery tube. There's air in the delivery tube, right? That get pushed through. So it might not be just oxygen going in there. It could be other gases like carbon dioxide or air that is being pushed through here or pushed through. Also, some of our oxygen may escape if the bung is not correctly put in. It's not a perfect vacuum seal kind of thing. Some of the oxygen may escape, so it may not be equal. So those would be my reasons. Some oxygen escapes from the bung, and some of the gases entering the delivery tube, the measuring cylinder and delivery tube, are not oxygen. So that would be my 2 marks. There's probably a couple of other things we could waffle on about there, but let's find where the answer was. Oh my gosh, zing to zing. So what was our answer again? Some oxygen may escape through a leak in apparatus, such as bung, not tight. Yeah, so some of the oxygen may escape. So what was the other thing we said? The other thing, my writing's coming out super small here. The other thing was some of the gas entering the measuring cylinder might not be oxygen, e.g., could be air or carbon dioxide from, was it a flask? I think it was a flask. I've even forgotten, from the flask or delivery tube. I mean, if the bung is not on correctly, it could be just air coming in from the bung as well, as we've stated in the first point. So there we go. That'll get you two fat marks. Right, let's move. Oh my gosh. 17.2 shows a graph of the students' results. So here we go. The volume of oxygen produced. 2AU is this one with the squares. 8AU is the one with the kind of whatever they're called, that look like diamonds. And then these little crosses are 20AU, the highest concentration. Explain the results for 20AU. How many marks is that, Bob? That's 3 marks. So we need to say kind of three things. So the way I would do this, you want to explain the results. So check out the different points. It's at its steepest here. Why are the results at their steepest there? So I'm going to say initially rate of reaction highest steep part of line. That is some bad English. So look how I spelled initially. Okay, the initial rate of reaction is the highest. And why is that? That's because there is more substrate available for collisions with active site of catalase or enzyme. So more enzyme substrate complex. I'm writing shorthand. Don't take your chance in the exam. Make sure you type it out. Enzyme substrate complexes um, are formed. Oh my gosh. So essentially, we've discussed and explained why the line is steepest there. We have the largest amount of products being produced because there are more enzyme substrate complexes forming, because this is the point of the reaction where we have the highest amount of active sites and substrates available for collision. Right. Now, why does the reaction start decreasing basically over time? So we want to say over time, the rate decreases uh, as substrate is used up, less enzyme substrate complexes form, right? So we've got less substrate, so therefore we're going to get less enzyme substrate complexes form. If I wanted to go detail, I could just try and over explain it and say like there's less collisions between the substrate and the active site, da 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 da, and then essentially that is going to explain the rest of this as well. So, but we might also want to say that the rate of reaction is higher than the others. So we might also want to say the rate of reaction is higher than the other concentrations because overall there is more substrate available, and so more oxygen will be produced. So let's say, so volume of oxygen produced is greatest at 20.AU. Nearly missed this out. So the volume of oxygen produced is greatest at 20.AU as there is more substrate available. So more product or oxygen produced. Okay, so there's more gas produced because there's more substrate. So I think we've covered everything there. Bish, bash, bosh. Right, enough of that. Let's move on. Oh, my days. Is that the end of the quick question? Explain the results. Yeah, me thinks it is. Okay, there we go. Okay, welcome to Let's Get to the Marks, going through question 18 of the OCR Biology A-Level, paper 2 from 2023. And this one's on immobilized enzymes. So why don't you read through the question, press pause on the video and have a go, and then unpause and watch the walkthrough. Let's go. So 18.1 shows three methods for immobilizing enzymes. We have adsorption, where the enzymes are stuck to a surface, covalent bonding, where they're bonded to a surface, and entrapment in a matrix. That's the enzyme molecule there on the key. State one other method for immobilizing enzymes. So there are many methods for immobilizing enzymes, and you do have to know some of the different methods. And I'm going to go through the answers. So what you have here is encapsulation. And encapsulation is where you take your enzyme and you trap it within like an alginate bead. And essentially, the enzyme here, this acts as a semi-permeable membrane, or it can be inside sort of an alginate bead, which acts as a semi-permeable membrane, allowing waste products to exit and resources, substrates to enter. And also, the fact that you can put a semi-permeable membrane around it means that it doesn't get detected by white blood cells if it's in the body, and it's not rejected by the immune system. So this version, we're going to say encapsulation, oh my days, encapsulation, or we can say in a oh my God, I can't type, semi-permeable membrane. And here we go. That's going to get you all of the marks for that one, which is just 1 mark. So you do have to know about all these different ones. Like covalent bonding is kind of cool. What happens is you covalently bond the enzyme to a surface, And what happens is this increases the stability, so the enzyme can now operate at a wider pH range. So if the enzyme, say, its optimum pH was pH 6, by covalently bonding it, you might now expand that range to it operating at pH 5, pH 6, and pH 7. So you can expand the pH range. You can widen the temperature range, so maybe it would normally denature after 40 degrees. However, maybe now it's more thermodynamically stable and will only start denaturing after 50 degrees. So therefore, you can use high temperatures, increase your rate of reaction. You get the idea. Entrapment in a matrix, very similar. You increase the pH stability and thermo stability, all of that kind of stuff. All right, let's move on. Also, technically, they're very good for reusing. So if you've got substrate and you want to pass the substrate across the enzymes and turn it into a product, so you turn your substrate into a product, or you push your substrate through this matrix and turn it into a product, it's very easy to get a pure product because the enzymes and the product and the enzymes are separated. Whereas if you just put them in a beaker or a container, you're going to have to separate the substrate and the product after. So you've got a purification problem. So these entrapment or immobilization of enzymes really help with making the purification process between the product and the substrate and the enzymes a lot easier. One advantage of using immobilized enzymes is that the enzyme can be reused many times. However, the process of immobilization can reduce the activity of the enzymes. This is a disadvantage of all immobilization of enzymes. With reference to figure 18.1, explain why the activity of immobilized enzymes might be lower than that of enzymes that are free in solution. So, let's have a go at that question. And essentially, if your enzymes are like in a matrix, like so, maybe their active site is not available for the substrate. So normally the substrate's going to fit in there, but maybe the active site of your enzyme is like facing the wrong way from which the substrate is going, and so you're not going to get enzyme substrate complexes formed. Or maybe the attachment to the matrix actually alters the 3D shape, the tertiary structure of the protein, and therefore it no longer binds to the substrate, the active site, so you get less enzyme substrate complexes formed. Of course, that can happen as well when you adsorb the enzymes to a surface. If it's not done carefully, these are perfect, but if it's not done carefully, you might end up with this situation where the enzyme's active site is not available to join with the substrate. So again, that is a problem with enzyme immobilization. If not done carefully, you may actually alter the shape of the active site. So the active site's shape may alter, or the active site, the availability of the active site may be reduced. And so that would be our problem. When free in a solution, so imagine if your enzymes are just floating around in a solution, they are going to be much more readily available to mix with the substrates. They're not locked into position, so they can't be locked into the wrong position because they can just rotate and get themselves into the correct position at some point. So that's why. Let's put that down into writing, shall we, and pick up the 2 marks. So immobilized enzymes may have reduced active, oh my days, site availability. Therefore, less enzyme substrate complexes form. So that's one possible reason. Another possible reason is the active site may be altered in 3D shape due to immobilization. So due to the adsorption or the entrapment within the matrix, where it's formed bonds with the surface, this may alter the 3D shape in the active site because, of course, that involves covalent bonds, ionic bonds, and hydrogen bonds and hydrophilic, hydrophobic bonds. So if you're forming other bonds, that may alter that 3D shape. The active site may be altered due to immobilization. Again, this may make enzyme substrate complexes form less readily or reduce the amount of enzyme substrate complexes forming. So anyway, there's some suggestions for an answer for that one. Bish bash bosh. It was only 2 marks. Don't ask me why I put 3 ticks. There we go. Let's go. Where are we? Invertase is an enzyme that catalyzes the following reactions: sucrose to glucose and fructose. Invertase is present in cells of Baker's yeast, a type of fungus. It is possible to immobilize invertase, and it is also possible to immobilize yeast cells. Suggest one reason why using immobilized yeast might be more expensive than using immobilized invertase. Why don't you pause the video, have a go, and then unpause. Okay, so what you've got to ask yourself, like, if you could immobilize the yeast, which obviously is a fungus that can speed up reactions, or you could immobilize, you could use an immobilized enzyme. So either immobilized yeast or immobilized enzyme. They both can do this reaction and speed up that reaction. So why might it be more expensive to use yeast? Think about what yeast is. It tells us what yeast is. Yeast is a fungus. And what does that mean? Well, it's a living thing. Although enzymes are biological molecules, they're not actually alive. They don't respire. So yeah, living thing, or biology, we call them an organism. So let's put that down. Yeast is an organism. Oh my God, an orgasm. No, did he? Right. Yeast is an organism, therefore needs metabolites to stay alive and function. So you don't need to supply your enzyme with glucose and oxygen for respiration and amino acids for growth and repair, but your yeast, you are going to need to supply it with metabolites to keep it alive, and that's going to mean it's more expensive and probably slightly more difficult to use yeast instead of an immobilized enzyme. Okay, let's zing on. Okay, investigations involving immobilized enzymes can be carried out using equipment like that shown in figure 18.2. A glass column with an outlet at the bottom is filled with alginate beads containing immobilized enzymes. So we've got the old encapsulation method here in the alginate beads. The substrate can be added at the top, and the product collected at the bottom. Actually, it doesn't have to be encapsulation. So this could be the alginate beans are acting as a matrix and the enzyme is just immobilized inside it. It doesn't have to be a semi-permeable membrane, probably not. Let's zing down. A student wanted to compare the ability of immobilized invertase and immobilized yeast cells to hydrolyze sucrose. So essentially, we're pouring through sucrose, and then out the bottom, we're getting our product, which should be the fructose and the glucose. So it's getting poured through like that, the sucrose, and as it drops down, the substrate, the sucrose, enters the active sites of the immobilized enzymes, and then out comes our product. So this is great because what this does is this means that your enzyme is separate from your product. You might get some unreacted substrate, some sucrose coming through, but your glucose, fructose, and any unreacted sucrose come through here, but they don't have enzymes mixed with them. So you can just take them back and pour them through again until you've pretty much ensured that all of the sucrose is broken down and you haven't got issues with separation of the enzymes from the actual product. You haven't got to do that, which you'd have to do if you had them all in a giant container. You'd have to, at the end, separate the enzymes from the product. This is like a continuous process as well. You can just keep pouring it down, pouring it down. It's not a batch process where they're all in one container and then you've got to purify at the end. Okay, let's go. Oh, this looks like a fat question. How many marks is that? Oh gosh, 6 marker it's going to be. A student wanted to compare the ability of immobilized invertase and immobilized yeast cells to hydrolyze sucrose. The student had access to the following: the sucrose solution, our substrate, alginate beads containing invertase, alginate beads containing yeast cells, glass columns such as the one shown, standard laboratory equipment and reagents, outline a valid method the students could use to compare the activity of immobilized invertase and immobilized yeast cells. Okay, so it's a 6 marker. What we need to do is discuss your independent variable, what you're going to change, what you're measuring, the dependent variable, and how you're going to measure it, what equipment you'll need to measure basically the rate of reaction for those two. And also, we need to talk about the control variables, and that should wrap up all your 6 marks. So try and break it down into that. Independent variable, how you're going to do that, what concentrations, etc., you're going to use, whatever. Dependent variable, what you're measuring, how you're going to do that, and then control variables, what you're going to keep the same. Why don't you pause the video, see if you can scribble those down, and then unpause and watch me go through the explanation. So the way I break this down for you guys, I'll bust out the IV, what you're going to do with the IV, then what you're going to measure and how you're going to measure it, and then control variables. I'll just roughly go through it. I can't really type that quick. Right. So what I want to do is we're going to want to set up like 2 columns, one column with the yeast in. Oh my days, check out the GCSE level drawing, wicked. And then we want to set up one with the invertase enzyme in, and we're comparing which one breaks down sugar the most. And what I'm going to probably do is also set up a control variable. And you might say, well, what's going to be in your control? Well, you just want to put alginate beads in that and prove that alginate beads themselves do not break down the sucrose and turn it into fructose and glucose, because then you've got an extra third variable in there. So we just want to prove that it's only the yeast or the invertase that's actually causing the breakdown of the sucrose. So let's put that down, and that's my first independent variable. So set up 2 columns, one with invertase and another with yeast. Lovely bubbly. There we go. A third with just alginate beads as a control. What I should do, set up a column, one with invertase immobilized and another with yeast. Hopefully, the examiner might get a bit funky and say you didn't say the immobilized ones, although that's what we're supposed to be investigating. So there we go. That's my independent variable covered. I want to have 2 columns, one with invertase, one with yeast, and then a control variable just to prove the alginate beads are not breaking it down. I might not get all of the points down because I'm having to do this on super fast. Okay, now what do we need? What else do we need? We could talk about the control variables here. You might want to naturally flow into the control variables. You might want to say something like, so I'm going to naturally flow into the control variables and skip the dependent variable. No, let's do dependent variable. Okay, so we can measure the speed of reaction using Benedict's solution, which will go from blue to a red precipitate, right? So with the presence of glucose, or with the presence of simple sugars, with the presence of reducing sugars like glucose that are produced as a product. So we can essentially measure the amount of glucose produced as a product when the sucrose is broken down by using Benedict's solution. And again, you could either use a known color chart of different concentrations and compare to your results. So essentially, you can compare the level of blueness that's left in your Benedict solution to a known color chart, or the level of red to a known color chart of different glucose concentrations that you get with the same volume and concentration of Benedicts in the same situation. And you can actually work out what is your percentage of glucose that's being produced and prove that either invertase or yeast produces more glucose. Or we can or use and I would go for either or, I wouldn't do both, or use a colorimeter. Make sure you zero it first, then which you do with a curvette and distilled water, and then or whatever, then record the transmission. Some people do absorption, so you could do emission, transmission, or absorption. Either one is good. Yeah, make a calibration curve, then read your results off. So there we go. That's explaining. So really what I've done here is cover the independent variable, what I'm going to change. Now I've covered how we're going to measure it, like the dependent variable. We're either going to use Benedict's, and we're going to check the color change against a known color chart, or we're going to use the colorimeter. First, you're going to zero it in a curvette, whatever. Then you're going to make your own calibration curve using 5 known concentrations, etc. And then you're going to plot that calibration curve and then read your results off to fairly accurately estimate your concentration of glucose that has formed. And so that's that. Now we need to talk about the control variables. Now I could have wrapped the control variables in all of this, so I could have talked about the control variables here. What do I need to keep the same about the yeast and invertase? So if I'm doing my control variables, I need to keep same volume of yeast and invertase, same number and volume of alginate beads. So what they're being immobilized on, we want the same number of that. We're going to want same concentration and volume of sucrose, right? So the substrate needs to be the same. It's not fair if one of them gets like double the concentration, then it's going to produce more glucose than the other. So same concentration and volume of sucrose. What else? What else? What else? Same amount of immobilization, the alginate beans, the kind of matrix or whatever that we're trapping it on. Same volume of yeast, same volume of the enzyme invertase. What else? Same concentration. I probably should have said same volume and concentration. So same volume and concentration of yeast or invertase. All of my days. Why is there a space there? Let's get rid of that. Trying to think, what other control variables do we need? Oh, what else is going to affect enzyme activity? Temperature. So same temperature. You could say use a water bath, use whatever. Same pH. So use a buffer solution to keep the pH the same. If you want to talk about the laboratory equipment, you could say use a buffer solution, same temperature, use water bath. Essentially, all of those are our control variables. And once we've gone through all of those, I think that's about it. Again, we might want to say, if we're using Benedict's, same concentration of Benedict's. And again, if you're going to say same concentration, you might want to say same volume as well. The worst thing they're going to do is say ignore that. They're not going to reject for you saying that. So same concentration, same volume of Benedict's as well. Now that should more or less cover all of it and get you all the fat 6 marks. So let's zing on. Now, as I said, one last thing, you could include the control variables within your discussion of the independent and dependent variable. So when you're saying set up your column with invertase immobilized and the second one with yeast, you could say make sure you use the same volume and concentration of yeast and invertase and the same number and volume of alginate beads. So you've kind of covered the control variables within your discussion of the independent variable. And then you could say set up the control with the same volume of alginate beads, but no yeast and no invertase. So I didn't really make that clear there. And then here we can measure the speed of the reaction. We then talk about the Benedict's. You'd say use the same volume, same concentration of Benedict's, etc. Use the same concentration of sucrose as a substrate. I didn't even say pour your sucrose through as a substrate. And then, yeah. So you're going to pour in the same volume and concentration of sucrose and let it trickle down. All right, guys, that's enough of that business. This is going to get you all of the marks. Make sure you like, share, subscribe, all of that business, punch up the bell, whatever. Let's get to the marks.
You're still here? It's over.
Okay, welcome to Let's Get to the Marks. This is an A-level biology question on the immune system. So 19. Animals are protected from infectious diseases by their immune system. Influenza is a viral disease that is common in winter months. It is transmitted when an uninfected person inhales droplets of moisture that an infected person has exhaled, often when coughing or sneezing. Explain how mucous membranes make it more difficult for the influenza virus to enter the body. All right, guys, basically a lot of waffle here. Really, it's just essentially saying, how does mucus deal with pathogens. And that's the translation for that question. 2 marks. Why don't you pause the video, have a go? All right, I'm going to go through the answers. So what you really want to know, you might be thinking, oh, do the mucous membranes make the diffusion pathway wider? Do they block the receptor sites so the virus can't attach and all of that? But actually what they want you to do here is just a GCSE level explanation of mucus. So mucus membranes obviously are coated in mucus, and that means they trap the virus. Then ciliated epithelial cells or cilia waft the mucus in up the respiratory system. Are we talking about humans here? Yeah, up the trachea into the mouth, and then it is swallowed and dunked into a bath of hydrochloric acid. So then the cilia waft the mucus, whoa, up through the respiratory system where it is swallowed and the virus destroyed. I think I'm overdoing it here in hydrochloric acid in stomach. So there we go. Yeah, swallowing mucus. Nice. Okay, so essentially that's going to get you the marks. Let's zing on. Okay, here we have a very, very common graph. If you don't know this graph, you need to familiarize yourself with this graph. You should have a revision card with this graph on, and you should know what all points and what's going on at the peaks and troughs on this graph. And it is the antibody concentration graph. That could be white blood cells or memory cells written over there as well. But normally it's the antibody concentration in the plasma or the blood. And then we have the first exposure to an antigen or the first exposure to a pathogen, a virus or a bacteria or fungi, etc. And then we have the second exposure. So this is really showing you the primary immune response and the secondary immune response kind of, well, yeah, after a second exposure. Let's get into it and stop the whiffle waffle. Different events during these immune responses have been represented as areas on the graph labeled with letters A to F. So you can see we've got A, B, C, D, E, and F. Complete the table with a letter or letters, that's key there, to indicate where on the graph the event is represented. So we need to put one of these letters or more than one of these letters in each of these rows going down here. Again, you don't have to use all the letters or you can use them more than once. It's worth 5 marks. Why don't you press pause on the video and have a go, and then I'll go through the explanation. All right, let me go through it. So antigen presentation. What is antigen presentation? Well, remember your white blood cell basically swallows the pathogen in phagocytosis. It forms a phagocytic vacuole or whatever we call that. And then eventually vesicles come and they fuse with that and they release their hydrolytic enzymes, lysozymes, whatever, and they destroy the pathogen. Now, that white blood cell becomes an APC, an antigen-presenting cell. It presents some of the proteins of the pathogen, i.e. virus or bacteria, some of the receptor proteins or whatever, on the outside, and it becomes an antigen-presenting cell. However, the pathogen itself over here is also an antigen-presenting cell itself. So it can be either that, or it can be a white blood cell which has become an antigen-presenting cell. So antigen presentation, why am I waffling? Where does it occur over here? Well, the antigen presentation must happen at A. It's their first exposure to the antigen. So that is when the antigen is presented. So the letter we want to put here is A. That's when our antigen is first presented. Now notice this is time along the bottom here on the x-axis. We've got a delay before the amount of antibodies go up. And that is because we're doing a bit of clonal selection. We haven't found the right clone yet so that we can start that mitosis and differentiation of the plasma B cells to make all the antibodies. We haven't done that yet until we get to B. So clonal expansion, hopefully you can see here, clonal expansion means when are you doing mitosis and making lots of white blood cells? You have to have done clonal selection first before you do clonal expansion. So clonal selection is going to be expansion, sorry, is going to be B because that's the order we've got going on here. And also over here after the second exposure, why is the antibody concentration going up? Because we're expanding our army here. This is where we are making lots and lots of copies of those plasma B cells. We're expanding our clone army. So we're going to say it is B and D for clonal expansion. Now, clonal selection, where is it that the clone is selected? Obviously, various different T helper cells come along and they might have the wrong shape. That's no good. So this is like our THCs. Eventually, you're going to get the right T helper cell. Oh my gosh, that doesn't look like it fits. But you're going to get the right T helper cell that has it should be more of a triangle. Yeah, whatever. I got a D in GCSE nowadays. I don't know what that is in nowadays money, but it's not a good grade. So yeah, we're going to come across the right T helper cell, which has the right shape. That's the clonal selection. And that, guys, is going to be happening at A. That's when clonal selection has happened. Clonal selection is not happening at B and D because once you've found the correct clone, you're then expanding the clone. You're making copies of it. You're doing mitosis. So by the time we get to B and D, you have found the correct clone. So we're going to go clonal selection at A. There's no letter here for any clonal selection, so we're just staying with that. Now, when do we have high T helper cell activity? When are we going to get the most T helper cell? Well, once the T helper cell locks on, so once we have our antigen presenting cell and it's presenting the antigen and our T helper cell locks into that and it's the correct one, that's when that is going to become T killer cells, what else does it become? That's when it's going to activate B cells, and the B cells are going to become memory cells. They're going to become plasma cells, etc. You get the idea. There's loads of stuff going on from that. So the T helper cell is going to be huge in the clonal expansion areas. So B and D is when the T helper cell is getting busy, is in B and D again. Okay, and then where would we get the highest number of memory cells? Now, memory cells are long-lived plasma B cells, so they can make the correct antibody to lock on and destroy the pathogen. But these memory cells, when are they going to be at the highest? Well, we're going to trace the antibody concentration for that. Wherever we have the most antibodies must be where we have made the most memory cells. And so there's quite a lot of antibodies there, but there's even more at E. So E has got the highest number of antibodies. So we're going to logically say that is where there is the highest amount of B cells and B memory cells and B plasma cells going down. And that is going to get us, oh, yeah, baby, 5 marks. Right, let's move on. Whiffle, waffle, waffle. Now, I might put this video on high speed. You can slow it down if you want, but I might put it on. Otherwise, it's going to be too slow to actually revise with. Fragments of antibodies known as Fab fragments have a number of uses. Snake bites can be treated using Fab fragments. Antibodies specific to the snake venom are produced using donor animals, usually sheep. These antibodies are collected from the animal's blood and then digested to produce Fab fragments. The patient can be injected with these fragments after being bitten by a snake. So it's like an antidote. You know, when they in the movies, like, who's got the antidote to the poison? It's basically some antibodies that will lock on and nullify, neutralize those toxins. Name the type of immunity that this treatment gives the patient. So we've got several options. You've got, when it comes to types of immunity, by the way, why don't you pause the video, have a go on a bit of paper, see if you can work it out. You can just do this one in your head. You've got 2 words you need to write here, 2 names. So you've got the types of immunity. Hopefully, you know, immunity can fall into, it's going to be either passive or active. Okay, so immunity can be passive or it can be active. And what does that mean? Well, passive means this is when, yeah, basically, I'll just get rid of that business. Let's just do what active means. And you can work out what passive means from that. So active just means your immune system. I was going to say your body. Whenever you want to write your body, just say immune system. So that's the kind of language thing. Whenever you feel like saying your body, just write your immune system when you're doing with immunology. So your immune system can produce the antibodies. So this is where your immune system can make its own antibodies. That means it's active. If you're receiving antibodies externally, then it's passive. And then the other thing that we need to deal with, the other 2 key terms is, so immunity can either be passive or active. It can either be naturale or artificial. So to give you an example, passive and natural would be breast milk. So a mother's breast milk contains the antibodies. The baby cannot make the antibodies themselves. The very early on newborn baby can't make its own antibodies. So it's receiving antibodies in the mother's breast milk, and it's a natural process. It's not being artificially, the baby's not being injected with antibodies artificially. So artificial means it's come from an external source that is artificial. Anywho, what would we say with this? You're injecting these fragments. So this is going to be artificial. And is it passive or active? Can they make it themselves? It's going to be passive. So it's artificial and passive, this one. And there we go. Yeah. Passive, they can't make the antibodies themselves. They're being injected with the antibodies for the snake venom, and artificial because it's an artificial process. Identify 2 similarities and 1 difference in the structure between a Fab fragment and a whole antibody. So compare this to what you know about an antibody. Hopefully you remember what a real antibody looks like. I was going to draw it, but then we're just slowing things down. So I'd say similarities. We've got, look, this is very similar. We've got 1, 2 binding sites. So I would say we want to say there are 2 antigen antibody binding sites. So 2 binding sites. I'm just going to make this smaller. You should write the full thing. So 2 binding sites. How many polypeptides? There's 4 polypeptide chains. So I'm just going to list a load of them. Okay. So there's 4 polypeptide chains. You can count that, right? Exactly the same in a normal antibody. You get 4 polypeptide chains. You also get this hinge region. So we also have a hinge region. Variable region is the same. So they've both got a variable region, which is where you've got these antibody binding sites. Oh, they've got disulfide bridges. Both have disulfide bridges. Can't remember anything else. There's at least 4 there, right? So, you know, pick any 2 of those. Could say the variable region's the same, antibody, antigen binding sites, same number. Right, the differences, there's quite a few differences. So look at the thickness of these polypeptide chains. If this was a proper antibody and not a Fab fragment, you would have 2 heavy chains and 2 light chains. So normally antibodies have 2 heavy chains and 2 light chains. So, and what else? There's no constant region. Where's the bit like that you would normally get with an antibody? That's where it binds to the white blood cell. Where's the constant region which it needs to bind to the membrane of white blood cells? So I would say the other difference is antibodies normally have constant regions. So that would be my difference. And there we go. You just need 1 of these 2 down here and 2 of these, and that would get you the 3 marks. Boom. Hopefully that's the question done. No, no, no. There's more, there's more, there's more. Okay. This is a long ass question. Infectious disease caused by pathogenic bacteria. That means bacteria, not all bacteria are pathogenic. Some are really good or useful for us. Infectious disease caused by pathogenic bacteria can be treated using antibiotics. The first antibiotics became widely available in the middle of the 20th century, thanks to Alexander Fleming with penicillin. Explain why many varieties of pathogenic bacteria are now resistant to a range of antibiotics. So really, this is, if you don't know this, put it on the back of a revision card. You need to know about antibiotic resistance. And it is GCSE, so it was kind of higher GCSE, this question, but you can just keep it to that. And actually, this is one of those rare A-level questions where you score the marks just with your GCSE knowledge. So what do we know about why we get antibiotic resistance and why this has led to it? So antibiotics Antibiotics create a cell. No, come on, Tom. They create a selection pressure. So when you introduce antibiotics into an animal, that creates a selection environment or a selection pressure where previously loser bacteria that were not more successful than the other bacteria, if they contain an allele that gives them resistance, now become king of the castle or queen of the castle. So antibiotics create a selection pressure in which those bacteria with a random mutation that has given them a resistant allele survive, right? So they're more likely to survive. Now, do I need to write all of this? No, I'm longing it out. You can probably just waffle a bit and say by natural selection. So you could say antibiotics create a selection pressure, and by natural selection, this creates resistant bacterial strains. Boom, you'll get that mark. Or I could go into loads of detail and say, let me show you the detailed version, but you can just stream this down to natural selection just to get that point. But I'm basically explaining natural selection. So they then pass the resistant allele to their offspring by binary fission. And so a resistant strain is produced. So what do we mean by that? What happened to the non-resistant ones? The ones without the random allele, they all die out, and only the ones with the resistant allele survive, and therefore they come back with all the resources just for them and make a massive population of resistant bacteria. Now we can streamline this whole process, this whole wiffle waffle, as it's a 3-marker, you could streamline this down to just the term natural selection. Essentially, natural selection is, well, this is the explanation for the natural selection process. So you could just say antibiotics create a selection pressure, and due to natural selection, a resistant bacterial strain is produced, and that would get you all the marks. So I just thought, for those of you that want to know what is the natural selection bit, it's the whole bit of the random mutation, giving you a resistant allele, more likely to survive, more likely to breed, more likely to pass on the allele to their offspring. I mean, more likely to breed, with bacteria, they breed by themselves, binary fission, so more likely to survive and do binary fission. Okay, what are the other 2 marks then? Because this really is probably only going to get you 1 mark on the mark scheme for mentioning that antibiotics lead to natural selection producing resistant bacteria. The other 2 marks are that if as antibiotics are over prescribed, okay, they're not just over prescribed, but they're used in agriculture as well, used in farming, and used widely in agriculture. Agriculture means used widely in farming. Basically, people injecting their chickens and their pigs and their cows and things that they're farming for meat with antibiotics to prevent the spread of disease amongst their farm, so they get a higher yield and all of that business. Actually, that's a bad thing because of the overprescription and used widely in agriculture. This is why we now have a wide range of many varieties. So this means many varieties of bacteria have been exposed. I can't spell exposed. Agriculture exposed. I hate it when it does that. You hate it when you're typing and then suddenly it jumps back a few lines and does a little jiggy jiggy. Means many varieties of bacteria have been exposed to the selection pressure, basically, to the antibiotics. So if you never expose bacteria, a certain species, to this selection pressure, then you'll never create a resistant strain. The fact that they've been used in so many different animals and so widely across the world means so many different strains of bacteria have been exposed and therefore have developed resistant strains. Now, the other thing that is screwing it up is also patients do not complete the course, and this allows resistant bacteria to survive and pass on the allele. So when the doctor says make sure you finish your 7-day course, what you're doing on day 6 and 7, 5, 6, and 7 is killing those hard resistant ones off so that you don't produce a resistant strain. So it's good for you and good for everyone else that you finish the course. And there we go. That's going to get you all the 3 marks. Now I could have streamlined that massively into just antibiotics create a selection pressure by which causes natural selection, formation of resistant bacteria. Antibiotics are overprescribed and patients do not finish the course, and that would get you the 3 marks. I've over-explained it just for your purpose, whatever. Plants and microorganisms have traditionally been used as sources of new medicines. Synthetic biology biology could play a major role in the development of new medicines. Outline how synthetic biology can be used in the provision of new medicine. So just give an example of how we use synthetic medicines, synthetic biology to make medicines. So let's look at insulin is the most obvious example. So genetically modified bacteria um, are transformed with the human insulin gene, which produces the useful hormone insulin for the treatment of diabetes. And that's going to get me all of the marks. Now, that's where they use those restriction endonucleases to cut out the insulin gene. You've got to identify it and all of that. And then they cut out the insulin gene. And then they use the same restriction endonucleases to cut, say, a plasmid. They insert with the ligase enzymes. They insert that insulin gene into the plasmid. And then you've got to transform the bacteria. You've got to get them to take it in using the shock electroporation or whatever. They get them to take it in. There's many different methods on that. And then identify the bacteria that have been transformed and then use them in fermenters to grow loads and loads of the bacteria, all producing the insulin for us. But this is just a shortened way, just saying use genetically modified bacteria, have an insulin gene inserted into them, and then they will produce the protein insulin for the treatment of diabetes. And there we go. That's going to get us all of the marks. Don't forget sharing, caring, hit the like, punch up the bell, subscribe. Let's get to the marks. Okay, welcome to Let's Get to the Marks. Going through this question. This is question 20. Grey wolves become whoa, rewind. Grey wolves became extinct in an area of North America in the 1920s. In 1993, some grey wolves were reintroduced into the same area. Grey wolves feed mainly on large herbivores, such as deer. The graph shows the population of grey wolves and some other species since the reintroduction. So we've got grey wolves here. They've got the straight line. We've got the dashed line is the deer, and the little triangles on a dashed line is the beaver colonies as well. Okay, so populations and the year at the bottom, and then we have each one of these representing those three different populations. Describe the changes in the deer population since 1993. So that we want to go describe the changes in the deer population. It's only a description. We don't need an explanation talking about resources or lack of predation and all of that. That's explanation. We just need to describe this line right here. Okay, that's the deer population, obviously times 1,000. So we're looking at 60,000 roughly at the start. What's the overall trend? You can see that yourself. There is an overall downward trend. So we want to say general trend is a decrease over time. So that's going to get us a mark for just describing the general trend. There's a decrease. Now we want to get a bit more nuanced. So we're looking like between 1993 and, oh my gosh, what is that? 1994. So there is an increase. So I want to say between 1993 and 1994 increases. And then let's give some other dates and deadlines, peeps. So we could also say that then between 1994 and what's that? 1996? No. 1998. Oh gosh. Yeah, between 1994 and 1998, we've got a decrease. So then decreases until 1998. Now, they just need to see a few of these numbers correctly given, and they'll award you for using the data. Okay. And then I might just go, I'm going to skip forward here. So I've just described a little bit more of the pattern. Basically, there are some fluctuations. So it fluctuates even though a general or overall downward trend. So although the population is going down, I'm proving that it fluctuates. I'm saying, look, it increases, then it decreases. So I'm showing that fluctuation. And then here you can see at the end here, what's this, 2006, because each of these two little squares equals 1 year. So 2006 to 2010, I would say there are very little changes in the population, or it plateaus or levels out. So we want to say between 2006 and 2010, whoa, 20,000, geez Louise, 2010, the trend levels out. You could use the word plateaus or stabilizes, whatever. Yeah, we can see that as well. And we're giving the dates and the facts and the figures. And there we go. That's going to get you the 3 marks. So remember, with these kind of descriptions, give the overall trend for the first mark. Then describe any fluctuations, giving some numbers to back that up. You could say something like the steepest decrease, find out where the biggest decrease was between which 2 times. You could say that. And then any parts where it has leveled off or plateaued, there's normally somewhere on there. Okay, let's go. Let's move on. The questione is B1. Calculate the percentage increase in the wolf population between 1995 and 2003. So we want a percentage increase here. Let's go up and look at the data. And 1995 and 2003, what I suggest, and it's the wolf population, what I suggest is you pause the video, have a go, and then I'll go through the answers. Okay, so the question was calculate the number or the percentage increase in the wolf population between 1995, which is over here, and 2003. And we're looking at the wolves only, which is this line here, if you are wondering. Let me delete that, but you see, and we're looking at that life. That's the wolf individuals. So this is individuals, so it's not 20,000. Our initial wolf population is 20. So we want to say initial wolf pop equals 20. What was our final wolf pop? Well, we're going to go to 2003, which was, we're going between 1995 and 2003, and it's there. We're going to zing across, and it looks like that is banging between 80 and 100. So we're going to go, our final wolf pop equals 90. Now, I know what you're saying. It's not the final wolf pop, whatever that is, but no, it is according to my question, because I'm looking at the percentage increase between 95 and 2003. Now, what is the percentage increase? Well, what you do, for the percentage increase, you want to do the difference or the change, you might prefer the word change. You want to do the change divided by the original times 100. And it doesn't matter whether you're doing percentage decrease or increase, that is what it's going to be. So what we're going to do, we're going to go 90 minus 20, and we're going to do that, which is 70. And we're going to do that over the original, which is 20. And then we're going to times 100. So let's smack that in the old calculator. So 70 divided by 20 equals that times 100. That's 3.5. And that is going to equal 3.5 times 100. We're looking at 350%. And that is going to be an increase, not a decrease, obviously. If it was a decrease, would have a negative in front of it. And there we go. That's going to smash all the marks. Boom. All right, let's zing down. Um, Between 2003 and 2010, there were no significant changes in the climate of the area. So it's not down to cold, temperature, whatever. North America, and no disease outbreaks. So we're not looking at pathogens, COVID of wolves, whatever. So just explain one reason for the changes in the grey wolf population since 2003. So let's check it out. What happened after 2003? We've got a decrease, right? There's a general decrease. Look, this is my wolf pop. And after 2003, yes, it goes down and up, but there's an overall general decrease. So we've got a general decrease. What could be the reason? If they're saying it's not the temperature and it's not disease, what could be another reason? Well, let's check out the deer population. What's happening to the deer? Deer population is decreasing. So maybe the wolves feed on the deer, therefore they have less prey, less food source, so they're decreasing. And that's all we need to put in our answer. Yeah. So, and it asks us for one reason. So I'm going to say deer population has a general downward, incredible general. Deer population has a general downward trend, and so less prey, food for the wolves. Yeah, wolves starve. I guess wolves starve. Some wolves starve. That's going to reduce their population. There's less food to eat, right? You get the coup. Let's move on. Now we're on part C. This is one of them fat ecology questions. Beavers are mammals that live in water. They form colonies nesting under big mounds of wood, set as islands in an area of water. So essentially, we've got these beavers. They're living in the water. So here's my little beaver. Don't ask me why he's orange. And he's living in the water, but they make little islands of wood in the water, and they like to live on these islands of wood. Okay, cool. In 1993, there was one colony of beavers living in the area to which the wolves were reintroduced. So just why the population of beavers is measured in colonies rather than individual animals. Now, this is so why do they measure the population in beavers in colonies rather than individual animals? So rather than saying there are 17 beavers, they'll say there are two colonies of beavers, one of 10 and one of 7, right? So they form colonies, and the colonies live under the big mounds of wood. So the colonies live under there, but at any one time, the individual beavers could be swimming about. This is a little bit like bacteria, where you have so many millions of them, it's easier to count the colonies than individuals. You couldn't possibly count individual bacteria. So you could count individual beavers, but it would be hard. So I'm going to say hard to count individual beavers, as they could be anywhere in the water. You'd have to set underwater cameras up everywhere. And how would you know you're looking at a different beaver? It's not like counting organisms on the land, right? The water is a three-dimensional space where they could be in any layer of the water. So it's hard to count beavers, as they could be anywhere in the water. Easy to count mounds of wood. It's easy to count the islands and know that each island has a beaver colony on it. So it's easy to count mounds of wood as each, um, whoop, yeah, each has a beaver colony attached. I don't really like beaver colony attached to the wood. That's not the word I was looking for. Each one houses a separate beaver colony is kind of like what I was looking for. Okay, anyway, there we go. Bish, let's get on with the next question. Scientists have claimed that the reintroduction of wolves has caused the beaver population to increase. Evaluate the support given by the evidence in the graph for the scientist's claim. It's worth 4 marks. So let's check out the graph and see that is it true that the wolves have caused the beaver population to increase? Let's evaluate the evidence and then comment on it. Let's go check out the graph. Let me delete my incredible drawing. Bosh. Here we go. We got the graph in front of us. So let's have a little look. Let's check out the beaver. No, did he? And the wolves. So we got beaver here. What can we say? From 1999, I would say the beaver population's going up from 1999. So beaver general trend going up. General trend of the wolves. Yeah, I mean the wolves were reintroduced in 1993, and their population's gone up, and the wolves have gone up. The beaver population has gone up after the wolves, so it might trigger. Look, the wolves went up first, and then the beaver went up after. So you could say that the increase in the wolf population has caused an increase in the beaver population. So that makes sense. Okay, that makes sense. Let's go put that in. That's support. What's the downside? Just because the wolves have gone up, it doesn't mean that they are directly causing the beaver population to go up. It might be a correlation. A correlation might be that something in the environment that enables the wolves to go up is also enabling the beavers to go up. And there is actually no causal connection between the wolves and the beavers. They're both going up because of a different variable. And we don't know that. So we can just say this is a correlation, not a causation. And that should give us all of the marks. Let's go down and put that together. So what we want to say, evaluate the support. We would say both the beavers and wolves have shown an upward trend in population. So that would be some evidence, and I'll give myself a little pat on the back for that. Give myself 1 out of 4 marks. And then I might want to say there was a time lag between wolf increase and then followed by beaver increase. So that kind of to me says, yeah, look, there was a little delay, which shows that the wolves were having an effect. However, what else do we want to say? Beaver and wolves do have different curves. So this would be this does not support scientists. So let me explain that one. I need to zing back up just to explain that to you. And what I mean is there anywhere we can see where the wolf population is decreasing and the beaver population is still going up? Look here. Oh, that's some crusty stuff, man. Between 2000 and 2005, the beavers are going up. But or let's say, between 2003 and 2005, the beavers are going up and the wolves are going down. So that would be my evidence against. So that would be some data that I'm using to support and against. Beaver and wolves do have different curves. This does not support the scientists, as I'm going to give some data. Between 2003 and 2005, wolf population decreases and beaver population increases. So you've always got to use some data, and there's my piece of data for this in these kind of things to back up your argument. So they go against each other. And the other final thing is we've got no causation. There is no suggestion of how the reintroduction of wolves is causing the beaver population to increase. So we want to say the graph just shows a correlation and not a causation. So that's the other thing. There is no causation shown by the graph, by the data. So we have no direct link between the wolf population and the beaver population, like a scientific explanation for it. Anywho, let's move on. I'm zinging down here. Oh my God, does this question ever end? Beavers build dams in water using trees they have cut down. Deer damage trees while they are feeding. Wolves often hunt deer when they are feeding near water. Oh my gosh, what is that all about? The following explanation for the increase in the beaver population has been suggested by scientists. The presence of wolves cause the deer to avoid grazing close to the water. This means the trees near the water are not damaged. Beavers have a plentiful supply of trees with which to build dams. So just one other piece of useful evidence that could be measured that would support the scientists' claim. Um, so we need to gather evidence. The presence of wolves causes deer to avoid grazing close to water. So I would say I need to know, where do the deers graze? So how long each day, um, as a percentage of grazing time, do deers spend close to water? So what I mean by that is, say they spend 8 hours grazing a day. I don't know deer habits, but if they spend 8 hours grazing a day, do they spend 2 hours grazing near the water and 6 hours inland? We need to know so we can say, look, when there's no beavers, they spend 2 hours by the water, and when there are beavers, i.e., or sorry, when there are wolves present, they only spend 1 hour close to the water. We need evidence to show data on how much time do they spend near the water. And then we could also look at um, tree damage, I guess. We could look at the tree population, like near the water. So another possible answer would be number of undamaged trees near water. So that's something else you could look at to back. You want some data of damaged trees away from the water and near the water. So, but either of these could get you the mark. Right, let's go on. Beavers are a species that was once native to Britain. The species became extinct in Britain about 400 years ago. In 2009, a population of beavers was brought from Norway and released into the wild in Britain. Explain why the reintroduction of beavers to Britain is an example of conservation. So, basically, because it's been done by humans, and that is our reason, because humans are trying to return the ecosystem. Now that sounds like, so conservation is done by humans, right? So conservation is done by humans. Um, in this case, they are restoring an ecosystem that they drove to extinction. So the humans are repairing something that they did. They drove, they're restoring an ecosystem, um, as in the population of beavers that they drove to extinction. Okay, so they're restoring the ecosystem to its original state. Now, conservation, conserving, keeping the same, could have been that before the beavers were made extinct, they introduced measures. So again, humans intervening in the ecosystem or the habitat in order to keep the beavers from going extinct. But 400 years ago, they didn't have the means, or they weren't bothered to do that. So suggest a situation in which the preservation of a habitat would be necessary. So just make a suggestion. In what situation would you need to step in and preserve a habitat? So this could be if a new disease was causing destruction to a habitat, e.g., a disease killing trees. So you want to step in and protect those trees to stop the habitat from being destroyed. So anything suggesting damage to a habitat or to a population within a habitat. It could be a disease ravaging one of the keystone species within the habitat, something like that, or a new predator has been introduced. Right, let's zing down. Oh, that's the end of that question on the ecosystem. Okay, question 21 is about the nitrogen cycle. This is from the OCR Biology paper 2, 2023, A-level biology. Let's go. The element nitrogen is recycled within ecosystems. Complete the sentences using the most appropriate terms. When animals die, microorganisms convert nitrogen-containing molecules in the animals' bodies to ammonia in a process known as boop. This ammonia is converted into nitrites and nitrates by boop bacteria. The bacterium boop converts ammonia to nitrites, and the bacterium boop converts nitrites to nitrates. Nitrates can be absorbed by plants and converted to boop. Nitrogen gas is very unreactive, but it can enter the nitrogen cycle by being converted to ammonia by boop bacteria such as rhizobium. Why don't you press pause on the video, grab your pen, grab your paper, have a go, see if you can write down the 1, 2, 3, 4, 5, 6 words that are missing and see how many marks you get. All right, I'm going to go through the answers. So hopefully you paused and had a go yourself, make the revision active. If you wrote it down or did it in your head, either way, all good. Let's go. When animals die, microorganisms convert nitrogen-containing molecules in the animal's bodies to ammonia in a process known as ammonification. That is right, ammonification. This ammonia is converted into nitrites, that's the NO2 minus, and nitrates, NO3 minus, by what type of bacteria? These are known as nitrifying bacteria. The bacterium, a specific name, converts ammonia into nitrites. Give me one sec, peeps. Crap's just popping up on my screen. Okay, and the bacterium, specific name, converts nitrites to nitrates. Check your syllabus for these because certain syllabuses don't need these. If you're doing AQA, you don't necessarily have to know, but I would recommend you do know. So the bacterium is nitrosomonas. And that converts NH3, which is ammonia, and it oxidizes it to NO2. You can see that there. It's added oxygen. That should be capitals. Never mind. And the bacterium that we call nitrobacter is also an oxidizer. It's going to oxidize NO2. And guess what? It's going to add an extra oxygen and make it NO3. So we're going to go from nitrites, NO2, to nitrates, NO3. Yeah, there's a charge on the electric charge as well, but you don't need to worry too much about that if you're a biologist. Nitrates can be absorbed by plants. Why do plants absorb nitrates? What do they convert them into? Eventually they're going to get converted into proteins. What are the monomers, the individual units of the biological molecule protein? So it is amino acids. So that's what we're missing there. Nitrogen gas, of course, those amino acids are joined together by peptide bonds. They form proteins and they're used for growth and repair. Hence, nitrates are very important if you want your crops to grow. So nitrogen gas is very unreactive. That's the N2. It's got a triple covalent bond, but it can enter the nitrogen cycle by being converted into ammonia by something bacteria such as rhizobium. So rhizobium, you find these in leguminous plants. They're found in the root nodules. Any idea what they are? They're called nitrogen fixers. So being converted to ammonia by something bacteria such as rhizobium. And these are nitrogen fixing bacteria. Okay, and so those are your 6 beautiful marks: one for ammonification, one for nitrifying, one for nitrosomonas, one for nitrobacter, one for amino acids, one for nitrogen fixing. That's a great little paragraph there to have on the back of a revision card that would help you with learning the nitrogen cycle. Right, use your knowledge of the nitrogen cycle to suggest and explain why plants do not grow well in soil with a low oxygen content. Look what we were talking about here with the nitrifying. What were we talking about? What's being added to the ammonia? Oxygen. What's being added to the nitrite to make nitrates? More oxygen. And without that oxygen, we can't make amino acids. And without amino acids, the plant can't make protein. And without protein, the plant can't grow. So you need to put that together into an answer. And it's going to go something like this. As it goes a little sign like this. Use your knowledge of the nitrogen cycle. So what we want to say is oxygen is needed for nitrification. Now, I could have said for nitrifying bacteria, but I'm just using the name of the process. So oxygen is needed for nitrification to turn ammonia. Oh my God, why is it called arsmonia? No, ammonia into nitrites and then nitrates. So that is a form of oxidation, right? So it's needed. Don't ask me why I'm writing in capitals. That's just annoying. My fat fingers are hitting the wrong buttons. Oxygen is needed for nitrification to turn ammonia into nitrates and then nitrates. Without nitrates, which is a soluble source of nitrogen, nitrogen plants can absorb in roots. They cannot make amino acids, so I'm longing this out, aren't I? They cannot make proteins, so they will grow less. Oh my days, did I long that out. So yeah, there we go. You basically, if you don't have oxygen, you can't do nitrification. That's a mark. And without nitrates, you can't make proteins, so you can't grow. So yeah. Let's zing. Is that the end of the questione? Now we've got a little bit more, tiny bit more. So rhizobium forms, where's my little red blob? Rhizobium forms nodules on the roots of certain plants. The rhizobium, it's a type of bacteria that is, receives a supply of glucose from the plant when the bacterium is which the bacterium is able to use. So look, they're living in a symbiosis, a symbiotic relationship where they get glucose from the plant and they help the plant absorb nitrates to make protein. So they're kind of I scratch your back, you scratch my back, kind of thing. So the rhizobiome receives glucose from the plant, which the bacterium is able to use. The diagram shows an incomplete diagram of the structure of alpha glucose. Write on the diagram to show the complete structure of alpha glucose, and that's 3 fat marks. So we've probably got to adjust 3 things. Use a bit of an exam technique. There must be 3 things we've got to add. Explain how the structure of glucose allows it to move from the plant to the bacterium. And so this is really testing us on biological molecules and then a little bit about transport. Right, let's go. I'm going to add the bits on, and guess what? I'm going to use pink. So we've got OHC. So how many carbons are in glucose? So just remember, you've got C6, H12O6, and you know that from GCSE. Use that as your checker to make sure that you've added everything in. So we've got 1, 2, 3, 4, 5 carbons. I need to add 1 more carbon in. Now, some people will make the mistake and add the carbon there. That's not where the carbon goes. If you know your structure of glucose, that is oxygen there. So your next carbon is here, and it's going to have a hydrogen, and then an OH group. And we can write that like that in displayed formula, or we can shorthand that and put CH2 and then OH. So you can do it like that as well. So I'm going to leave it like that. CH2OH takes up less. And then if you wanted, you could count up all the hydrogens and all the oxygens. Look, and make sure that you have enough. You've got 1, 2, 3, 4, 5. So I need 1 more oxygen, which means I'm going to need an OH. And if I was to add all of this up, I've got 10 hydrogens. 1 there, 3, 4, 5, 6, 7, 8, 9, 10. I need 2 more hydrogens. So I'm going to need an oxygen and 2 hydrogens. So the question is, do I do that? Or do I do that? Like that? Do I put the OH above da 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 da? Now, the way I see it is the alpha, it tells us that this is alpha glucose. The only difference between alpha and beta, I think of the B, meaning the hydrogen is below, and the alpha, the hydrogen is above. So actually, this is the correct way. Now, one other thing that you might want to do, because if you're dealing with like certain examiners are really particular and they say they want to see that bond in between the O and the H, and they don't like it when you write the O and the H together and they like it like that. So you can just try to be really careful. And there we go. This is alpha glucose. Again, I think of alpha, the A in the word alpha meaning hydrogen above, and the B in the word beta meaning hydrogen below the carbon. And that's carbon 1, 2, 3, 4, 5, and carbon 6 is outside the ring. Okay, explain how the structure of glucose allows it to move from the plant to the bacterium. So have a little look, peeps. What is that? That is delta H. That is delta negative. We've got the lone pair on the oxygen that can bond, form hydrogen bonds, essentially. So this is a polar molecule, meaning it has an electric charge. It has positive bits and negative bits. It has poles like north and south. And what that means is it can form hydrogen bonds with water. Therefore, it is soluble. And yeah, that's why, because it is soluble. So we just want to say that glucose is a polar molecule as it has slightly positive um, hydrogen and slightly negative oxygen. Now, I might be going over the top because I might just get them up saying glucose is a polar molecule. I'm pretty sure I will do. So hydrogen is slightly positive and slightly negative oxygen. These can form H bonds, and I probably should type out the word hydrogen just in case the examiner's being really strict. These can form H bonds with water. So glucose is soluble in water and can move yeah, it's soluble in water and it can move down the phloem. I'm going to be extra by mass transport to the roots where the bacteria live. So it's going to go from the leaves to the roots by mass transport, source to sink, on its way to visit the bacteria in the roots. And the reason why it can do that is because glucose is polar. So my suggestion, I haven't checked the mark scheme on this one, but this is definitely 1 mark for that, 1 mark for that, and 1 mark for that. And then down here, I would suggest the marks on the mark scheme should be, but you can never trust the mark scheme fully. Glucose is a polar molecule, and therefore it is soluble in water, and maybe saying H bonds, so soluble in water. I know it's 2 marks. I'm just trying to tick where the key bits are. I've gone a little bit extra by saying so it can move down the phloem by mass transport, because it does say how does it allow it to move from the plant to the bacteria. So, but I think really the fact that it's polar and it's soluble in water would be my 2 marks there. And that is the end of the question. Don't forget sharing's caring. Hit the like, punch up the bell, subscribe. Let's get to the marks. I'll catch you lot on the next vid.
You're still here? It's over.